Welcome back, everyone. Okay, today we're opening up the notes and diving into, well, probably one of the most specific documents we've tackled here. It's titled Notes on Fishkeeping, Part 143.
Right. And just to be clear, this isn't Fishkeeping 101.
No, definitely
Yeah.
not. This is really high-level stuff. We're talking about specialized sourcing, the kind of deep expertise it takes.
Exactly. It's less about the fish themselves, almost, and more about the intense criteria, the incredible patience involved when someone is hunting for one very specific high-end thing, like a single specimen in this case.
A very dedicated search.
Yeah.
So let's name the target here. We're focusing on the Turkana Jewel Cichlid. That's Rubricatochromis exsul for the scientists among you.
A beautiful fish.
And the author of these notes, someone named Ang, actually wrote they'd been passively searching for years, just years.
Yeah, that detail really jumps out. Passively searching. It tells you this isn't something you just pick up at the local shop. It's rare.
So years of looking.
And then, boom, Ang finds a group. Finally. You'd think that's the end of the story.
Right.
Success.
But nope. The notes immediately shift to why Ang rejected them. And that's where we get into these fascinating expert criteria. The why not is the real story here.
Okay, that is intriguing. Finding them was just the first step, followed instantly by this critical analysis. So what was Ang actually looking for? What was the ideal outcome after years of waiting?
Well, it wasn't just about getting the species. The goal, explicitly stated, was to start with an already bonded pair that exhibits vibrant color.
Ah, okay, so two key things there. Bonded pair and vibrant color. That's way more specific than just buying, say, a juvenile fish.
Miles more specific. A bonded pair suggests they're already compatible, maybe even ready to breed. And vibrant color. Well, that speaks to the quality, the aesthetics, the genetics everyone wants in a jewel cichlid.
So if the fish found didn't tick both those boxes.
Then, for Ang's purpose, they were essentially useless, despite being the right species after a long search.
Right, finding the fish versus finding the right fish. So the notes say the first big problem was visual. The fish were quite pale. Yes. And for a jewel cichlid, known for intense reds, pale is a huge warning sign.
It immediately throws things into question. Why would that paleness be such an instant potential deal breaker for an expert like Ang?
Because that paleness isn't just one thing. It forces the expert, Ang in this case, to consider three quite different possibilities. And only one is really okay.
3 scenarios from just observing pale fish?
Okay. It shows the depth of thinking. An amateur might just think, oh, maybe they'll color up. Ang is thinking environmental, genetic, behavioral, a whole decision tree.
All right, let's break that down. Scenario 1, you said it's the most hopeful one.
That's the environmental idea. Basically, the fish might just be stressed. They were recently introduced and still adjusting, as Ang puts it.
Makes sense. New tank, transport.
Yeah.
That washes out color in lots of fish, right?
Right.
Temporarily.
Exactly. Temporary stress. Give them a stable environment, good food, some time. And if the genetics are good, the color should bounce back. It's recoverable.
Okay, like cleaning surface dirt off something valuable, manageable. But scenario 2 sounds worse. How do you know it's not that?
Well, that's the risk. Scenario 2 is the genetic possibility. Ang considers this might be a rejected batch with naturally subdued coloration.
Rejected batch, meaning
Meaning, maybe these are the ones someone else already looked at and passed on because they're just genetically dull. They'll never achieve that stunning blood red color no matter how well you care for them.
oof. So investing in those would be a total dead end for the vibrant pair goal.
A complete waste of time and resources, yeah. That's a huge risk in specialized sourcing, mistaking a fundamental flaw for a temporary issue.
That distinction is critical. Okay, but there's a third possibility. You mentioned behavior.
Yes, and this one is, I think, really fascinating. It's about social hierarchy. Ang wonders if these fish might all be non-dominant or breeding males who haven't yet developed the stunning blood red color associated with the top fish.
Wait, so the color isn't just genetics, it's tied to status. Like, only the boss gets to wear the bright red tie.
Pretty much. In many cichlids, especially males, the most intense coloration is a signal of dominance. It says, I'm the toughest, fittest one here. I control this territory.
Huh. So if you have a tank full of pale males...
You might be looking at a group of subordinates. Maybe the dominant male was sold, or maybe they're just keeping their heads down, literally suppressing their color to avoid fights.
Wow. So they could be genetically capable of stunning color, but they're, like, actively choosing not to show it because of social pressure.
Exactly. They're socially conditioned to be pale. And if you buy that group, Well, how do you get one of them to step up and claim dominance, to show that color?
That sounds complicated. You'd have to change the social dynamic somehow.
Precisely. Maybe introduce a female, maybe another male, something to trigger competition. But that's potentially messy, could lead to aggression. Aang's goal was to avoid all that drama by getting fish that were already showing their quality, already vibrant.
Okay, I see the dilemma now. Is the paleness just temporary stress? Or is it bad genetics? Or is it this suppressed dominance thing?
And 2 out of those 3 scenarios mean failure if your goal is a vibrant bonded pair quickly.
The discipline to see that and to walk away from a fish you've searched years for based on that uncertainty, that's incredible. It's all about understanding the species' behavior.
It really is. And that decision to pass wasn't just about the color. There's a second major issue, moving from aesthetics to basic biology.
Okay, what was that?
Gender. Aang couldn't clearly identify any females in the batch.
Well, that kills the bonded pair goal right there, doesn't it?
Yeah.
If you can't even confirm you have a female.
Completely. No female, no pair. Simple as that.
But hang on, why would it be so hard for someone experienced like Aang to tell males from females? Are they really similar looking?
It can be, yeah. Especially when they're younger or stressed like these pale ones probably were.
Mm.
The notes mention the specific subtle differences Aang was looking for.
What are they?
Females typically have shorter fins and a rounder body.
Typically. Shorter, rounder. Those sound like subtle cues. Not exactly obvious differences.
They're very subtle. Think about it. If the fish are stressed, they might clamp their fins, hiding the length difference. Body shape can vary with feeding or condition too. You need clear, relaxed fish to be really sure sometimes.
It's like trying to verify something tiny on a blurry photo. You just can't be certain.
Exactly. And in sourcing, certainty is key. You don't want to gamble. You could buy 5 fish thinking you've got a mix and end up with 5 males. That tanks the whole project.
Okay, so putting it all together, the paleness raised flags about quality, maybe stress, maybe bad genes, maybe suppressed dominance, and then the inability to confidently spot a female made the core goal of a bonded pair impossible to guarantee.
Correct. The batch failed on both the vibrancy check and the pair potential check. So despite years of searching, Aang had the discipline to maintain those high standards and walk away.
Passed on the batch. Wow.
It shows that patience and sticking to your criteria are paramount. An impulsive buy, getting the wrong specimen, is often far worse in the long run than waiting longer for the right one, especially in high-end specialized areas. Time, money, effort, all wasted if you compromise too soon.
That's a powerful lesson, I think, for anyone listening, regardless of their field. Whether you're sourcing rare parts, evaluating code, or, yes, keeping high-end fish, that deep knowledge, understanding the subtle signs, the behavior, the morphology, it trumps the excitement of just finding something.
It really does. And, thinking about Aang's criteria, rejecting Marat for being pale, possibly non-dominant or genetically subdued, it raises a really interesting question for me.
Oh.
Well, Aang is clearly an expert, applying these very high aesthetic and behavioral standards. What does that imply about all the other Turkana Jewel Cichlids out there in the hobby? The ones that don't meet that bar.
You mean the pale ones, the ones that maybe are from rejected batches or are permanently subordinate?
Exactly. What happens to them? Experts are selecting for this vibrant, dominant ideal. Does that mean there's this whole population of, well, less spectacular fish circulating that don't make the cut for serious enthusiasts?
Huh. That's a really provocative thought. What's the fate of the aesthetically subdued or behaviorally non-dominant fish? They're still the same species, still rare maybe, but rejected based on these expert standards.
It makes you consider how much our human aesthetic preferences, our collecting standards, actually shape the populations within these hobbies. We create these hidden hierarchies, valuing only the best specimens. What does that do long term?
A kind of artificial selection driven entirely by the collector's eye. It's definitely something to chew on, thinking about those unseen hierarchies, not just in fish, but in any specialized collecting field. Fascinating.
Indeed.
All right, lots to think about there. Until our next deep dive, keep looking closely at those details and maybe spare a thought for the quiet ones.